compound bryostatin being used in over 1,400 patients. We have compassionate use data from several compassionate use patients where we have actually seen disease reversal in a severe Alzheimer's population. We have completed phase 2A. We have met all our primary endpoints, in addition to which we've actually seen target engagement where bryostatin had activated PKC epsilon. We are presently enrolling and dosing patients in a 2B proof of concept trial. We are studying 150 patients in 30 sites across the United States. And I think one of many things that makes us a bit different is that our study is dealing with a moderately severe to severe Alzheimer's population. In addition, and if we have time, we'll get into it just very briefly, we also are using bryostatin in several orphan disease programs, including Fragile X syndrome. It's part of the autism spectrum. We've been granted orphan drug designation by the FDA, and Niemann-Pick type C disease. Neurotrope was founded in 2013 to develop and commercialize bryostatin as a disruptive approach with the potential to reverse Alzheimer's disease. We licensed patented technology from the Blanchette Rockefeller Neurosciences Institute in West Virginia. At the time we came along, over $200 million had already been invested in this technology, about $100 million through the NIH, and in excess of another $100 million through the Rockefeller Institute in extensive preclinical work, which created the foundation for our clinical advances. To date in Neurotrope, we have $40 million of capital invested in our company. There is a lot of data on what we've accomplished, extensive publications in peer-reviewed journals. I ask any of you who may be a bit more interested to see us after the presentation. We can send you a lot of information, or please visit our website at Neurotrope Inc. or Neurotrope BioSciences, where there's a host of information available. I don't need to really review this slide telling you, we've heard it numerous times, I'm sure you all know it, about the tremendous need for something in Alzheimer's and what it costs our healthcare system. What are some of the milestones we've already achieved in Neurotrope using bryostatin? In 2013, we had our first FDA approval for compassionate use treatment. In 2014, we saw amazing beneficial results in our first compassionate use patients, and the FDA approved a phase 2A trial, approved our protocols. In 2015, we completed the 2A trial, achieved all our safety endpoints, no adverse events were reported. We showed target engagement in increasing PKC epsilon levels, and we continued our compassionate use program, continued to see tremendous efficacy. In 2016, we have begun dosing our first patients in our phase 2B proof of concept trial. And we'll speak a bit more a little bit later about what that trial consists of. And we expect our first interim data to be unblinded in the first quarter of 2017, about a year from today, and the full data completed within the first half of 2017. What is bryostatin? It's derived from a marine organism called Bugula neretina. We are really bringing to market pioneering science developed and published by our Chief Scientific Officer, Dr. Alcon. He's a former Chief Laboratory of Adaptive Systems at the National Institutes of Neurological Diseases and Stroke at the NIH, and he's the Senior Scientific Director of Bernie, the Blanchard Rockefeller Neurosciences Institute, holding the Toyota Chair in Neurodegenerative Disease. Bryostatin is a very potent PKC epsilon activator. It promotes synaptic function, synaptogenesis, degradation of amyloid B, and cognitive enhancement. It has potential broad application in other neurodegenerative conditions as well. At the present time, while our clinical trials are using bryostatin that's been extracted and purified from the natural Bugala neretina, we are in the process of synthesizing bryostatin for commercial use under a license with Stanford University in the laboratory of Dr. Paul Wender, one of the premier chemists in the United States. I would like to, if I could now, Dr. Alcon, if you don't mind, speak for a few minutes on the scientific principles of PKC activation. Thank you.
Thanks. So just to continue where Charles left off, and somewhat where I left off in my talk a couple of hours ago, we started studying the PKC epsilon cascade in normal animals in a broad range of species, from snails to rodents to rabbits, and looking for endogenous molecules that were activated during associative memory, spatial maze memory, olfactory discrimination, learning, etc., because we wanted it to be potentially generalizable to humans. And we saw that the PKC activation then became associated in the dendritic tree with new synaptic formation, which we identified with electron microscopic reconstruction. Once we saw that maturation of fully functional synapses under the electron microscope, it was a natural thing for us to ask to what degree this might be possible in the context of neurologic and psychiatric disorders. So we developed at the Rockefeller Institute 10 different animal models, 3 different Alzheimer transgenic models, the TG2576 single gene, the 5XFAD quintuple gene, and another double gene. In addition, we had 3 stroke models, and then a Fragile X mental retardation, etc. What's in common with these, and we talked about this earlier this morning, is there are, in common with all of these, a loss of the wiring in the brains of those animals. And I would suggest to you that that's in common with a lot of neurologic disorders that probably comprise about 7 or 8 million patients' disorders in the United States alone today. That includes Alzheimer's disease, includes stroke, and includes traumatic brain injury, and includes Fragile X mental retardation, the most common genetic cause of mental retardation in the United States, and a comparable number in Europe. They all have a loss of the wiring. And this is, if you will, the elephant in the room. This is the elephant that, to some extent, no one wants to see. When you're getting rid of amyloid, are you restoring the wiring? Are you bringing them back? Are you preserving the neurons from degeneration? PKC epsilon is not only synaptogenic, it's also anti-death or anti-apoptotic. It revitalizes neurons that are already dying, and we can prove that by using certain stains to show that the dying neurons are revitalized, as well as throwing out new connections. So it's rebuilding the wiring, and it's not rebuilding the wiring for a couple of weeks or even a couple of months, but we've shown that even learning information that was acquired 4 months previously, before a stroke or before an incident, can then be preserved by the bryostatin treatment, and even lasting many, many months thereafter. So it's a long-term treatment. We already know this. And this is also validated, or at least consistent with what we see in our compassionate use patients. The compassionate use patients gave us dramatic data, not only experiencing what was possible for patients, but also learning what doses would work. Fortunately, we're working and we're motivated the phase 2b trial designs with this data, but we're also optimizing the dosing frequency by learning what works for the compassionate use patients without causing toxic side effects. So the long-term nature of these efficacies in animal models at least resonates with what we've seen in compassionate use patients. There's one point I made earlier, I tried to make earlier in my talk this morning, I would like to reinforce it here, and that is, and this was somewhat fortuitous, but perhaps it suggests good thinking about the problem. If you consider that Alzheimer's disease is a disease of synaptic loss or synaptic dysfunction, not just not enough transmitter going across the cleft, but a disease that actually is killing neurons and synapses, and that it's a disease of the synapses that normally is tended to by a balance between A-beta oligomers that have a normal role in brain function, a balance between that and, I'm going to suggest, the PKC epsilon pathways, because those two are balanced normally. And that balance is maintaining a certain equilibrium of synapses in order for us to learn new information, even into our adult and aging lives, as well as in children. If you consider that it's a balance and that it's a synaptic derangement, then it's not surprising that if we learn how to restore that balance, we also may be able to address all of the other major consequences of the disease, such as amyloid plaques, A-beta oligomers, tau phosphorylation, all the things that you've heard about at this panel that have been going on for 30 years and have never led to a disease that stops the progression or treats the underlying disease. I'm sorry, but this is the truth. And if you listen to the panel, they were saying the truth, except that it was all mixed up very frequently because they didn't actually come out and say, we don't have a drug that ever addressed the underlying disease. And the point here is that if you are addressing something that is targeting normal balance of synaptic maintenance in order for the brain to register information, it's not surprising then that that normal balance is also going to involve the pathophysiology and the equilibrium of the enzymes that maintain the balance, such as those that, when deranged, produce plaques, or when deranged, produce neurofibrillary tangles, as well as degeneration. So it's not too surprising that this therapy that has gone through the window of synapse reclamation or restoration also addresses everything else about this disease, including even the risk gene, ApoE. I don't think it's an accident that ApoE4, the mutation, stops the PKC epsilon expression and stops, therefore, the synaptic formation, whereas the normal gene, ApoE3 and ApoE2, promotes that expression. And bryostatin directly interrupts the ApoE4 mutation. It's a kind of genetic therapy. So that suggests, and I'm going to try to end here quickly because I want Charles to have a few minutes of his own, but that suggests that not only can this drug platform, and it's a platform with a lead compound named bryostatin, but we have a platform of drugs, that this will address not only advanced severe Alzheimer's, which we've been doing in compassionate use patients, and now we're doing in our phase 2b, but also moderate and also early and maybe even pre-dementia patients, because they have appropriate targets in every one of those phases. And then I turn it back to you, Charles.
Thanks, Dan. Thank you. Well done. Thank you very much, Dr. Alcon. And I'd like to leave some time for Q&A because I'm sure we'll have some questions primarily for Dr. Alcon. But I just did want to conclude saying a couple of things. As Dr. Alcon said, no drug has ever been approved for slowing progression or curing the disease. Approved drugs modestly treat symptoms. What we are looking to do at Neurotrope is treat symptoms, slow progression, and reverse the progression. In summary, I'd like to just point to some of the, I think, highlights of what we've stated, but just to recap them. We're a late-stage biotech with bryostatin 1, a novel compound for Alzheimer's disease. We're a new paradigm. We're different than anything else that is out there. Our primary focus is synaptogenesis through PKC activation while also inhibiting amyloid plaque and tau. We're leveraging a $200 million R&D investment that was made in the past before our company was formed. We're dealing with pioneering science developed and shown by Dr. Alcon. We've demonstrated improvement in compassionate use cases, and we're currently enrolling and dosing patients in a phase 2B. The last thing I want to say is on a personal note. We just came back from our investigators meeting where we had healthcare professionals, doctors, nurses, and others from 30 sites all across the United States. And everyone was extremely enthusiastic and working not just like a team, but working like a family on a great mission and a great endeavor. And one thing that sticks out in my mind that I'd really like to share is one of the physicians who came up to me and said, you know, I have patients when they had early onset, moderate Alzheimer's, there were drugs I could give them. I'm not sure if they're working or doing anything, but there's drugs I can give them. When these same people that I've known for years come in and they turn severe, there is nothing I can do but watch them get sicker until they die. And he said, I am so rooting for this trial and so happy to be a part of it. We really need something because there's nothing out there. That's our mission at Neurotrope, and that's what we're hoping to accomplish. So thank you, and if anyone has any questions.